Okay, very good afternoon, everybody. Right. So today we discuss the plant metabolic. So plant metabolomics is the a wider topic. This one is a very wonderful lesson. This is the last session. So here the meaning of plant metabolomics. The metabolomic aims to quantify and identify all metabolites in a given biological context, an aim that admittedly is as challenging as full scale proteomics. Means here all the metabolites we have to calculate that one, the biological. So, so in this thus a manifestation of endpoint of metabolic and physiological process that taken individually measurement of metabolism in the different physiological conditions are likely to be more indicative for the purpose of systems biology studies than transdermos profiling. The metabolism is a complex set of metabolites in a cell or tissue, consists of low molecule, low molecular weight. So the chemical intermediates, which are the end product of gene expression. It is understood that any change in gene, either protein expression level, will have only small effect on metabolic fluxes. They must have a large effect on metabolite concentration. The metabolomic approaches seek to profile metaboloids in a non-targeted way, that is to effectively separate and detect as many metabolites as possible in a single analysis. Eventually, the information on transcript and protein bound abundance was combined, and this would identically ideally lead to nearly complete molecular picture of the state of a particular biological system at the given time. The plant metabolomics is essentially a comprehensive, high thought and analysis of complex metabolite mixture, typically of plant extract. To achieve the overview of metabolic composition, it is essential to establish a multifaceted, multifaceted, fully integrated strategy for optional sample extraction, metabolite separation, detection, and identification, automated data gathering system, handling and analysis systems, and finally quantification. Both here analytical and computational development are essential to achieve this aspect. And moreover, this data also is the required proper storage in suitably configured databases. It is very appropriate that the large quantities of metabolomics or metabolic profiling data, if generated, will help to open up many previous inaccessible areas of plant drug research. The objective of metabolomic targets assessing metabolic changes in a comprehensive and global manner in order to check the biological functions or provide the detailed biochemical responses of cellular system. The metabolomic system society means the metabolomic society in 2005 is to promote the international growth and development of field of metabolomics, to provide the opportunity for the collaboration among the researchers in metabolomics, including connection between the academia, government, and industry regulations and vendor in the field of metabolomics, and to provide the opportunity for a dissemination of research achievements in the workshop, conference, and other journal publications. The first International Congress and Plant Metabolomics was held in the Wageningen, the Netherlands, April 2002, with the primary goal of bringing together these scientists who are actively involved in this field and those who soon to plan to be in the future. So, one is the first gathering. In such instances, opportunities are credited for the collaboration and joint strategies can be determined to meet the metabolomic challenges. The selected abstracts from the oral and poster presentations at the meeting are now accessible at metabolomic, and the site will continue to be used as aid for the information exchange and collaboration. So, although microbes prove to be richest overall source of metaboloids, plants are the source of most complex individual mixtures. So, the Mariant van der Wef in the Netherlands reports that it has been predicted the bacterial genome, which were already been sequenced, can support the biosynthesis of just a few hundred. Sorry, guys. Sequence can support the biosynthesis of just a few hundred metabolites. Example 550 for the Bacillus subtilis and 800 for Escherichia coli. But for individual plants, this value is likely to be in the tens, uh, tens of thousands, number of thousands are there. Approximately 50,000 different phytoconstants have been illustrated in the plant, and it is the predicted that the final figure for the plant kingdom will approach or even exceed 2 lakhs. Is identified in 2001. Behem. Thus, metabolomic effect exerts as a considerable challenges for the plant scientists. The metabolomic information not only will assist in the deeper insights in understanding of the complex interactive nature of plant metabolic network and their responses to environmental and genetic change, but also will provide information regarding the fundamental nature of plant phenotype in relation to development, physiology, tissue identity, and resistance and biodiversity also. So the coupling of electrospray ionization with mass spectroscopy with electrophoresis and hydrophilic interaction of chromatography has been successfully used to analyze the metabolomic problem. So these are the some equipment we have to use to identify the metabolomic issues in the plant. The metabolome is a complex mixture of chemical compounds with the broadly differing the chemical and physical properties which presents the limitations to all the analytical technologies. Instrumentation utilized in the instrument of metabolome includes mass spectroscopy, fluorescent transformation, infrared spectroscopy, nuclear magnetic resonance spectroscopy, NMR, nuclear magnetic resonance spectroscopy, that one, NMR is also used. A number of analytical strategies are employed is ranging from the study of a few metabolites common to a specific metabolic pathway or chemical classes such as amino acids to the comprehensive analysis of hundreds of chemically diverse metabolites, that is including the metabolite to propylene, metabolic finger printing, and metabolomic disaster. Nowadays, means currently no single technique can be pro non-biased detection for all the compounds present in the metabolome, and in future application it is expected there is a number of different technologies will be employed to fulfill this objective. Metabolite profiling involves separation of metaboloids using the gas chromatography, liquid chromatography, or either capillary electrophoresis prior to detection of individual compounds and with metabolite identification by comparison to already available mass spectral or retention index libraries. So, however, for larger studies, sample separation and analysis are both time consuming the laboratories. The typically run times of 10 to 20 minutes gives the detection of more than 90 metabolite peaks are identified. To overcome the problems for long run times when the analysis large sample set, metabolic fingerprinting can be employed. This involves analysis at the expense of lower sensitivity and a reduced ability to identify metabolites. The objective is to provide high throughput analysis with the objective of screening samples to enable discrimination between the samples of differing biological origin or status. The plant metabolomics aims at providing comprehensive information that could help to optimize metabolic engineering strategies by providing more information about the biosynthetic pathways and the interaction between these. Metabolomics is the study of all the metabolites of given biological samples, but since no single technique proved for the comprehensive detection, numerous analytical tools such as the gas chromatography, liquid chromatography, means mass spectro, and nuclear magnetic resonance have been used to investigate the metabolome. So here the notable advance have been made with a single technique such as this means gas chromatography or either a spectroscope, mass spectroscope. Studied that involves multiple techniques such as NMR and liquid chromatography, ultraviolet chromatography, or mass spectroscope, spectrometry. MS means mass spectrum. Is estimating and monitoring the metabolome are perhaps two of the most important analytical challenges of the 21st century. While the methodologies for genomic and proteomic analysis have been uh, stabilized into several well characterized approaches, analytical methods for the metabolomic analysis remain in the considerable flux. The problem research with the entire diversity of the metabolome estimates for the number of metabolites in the human and plant metabolomic ranging which is from 2766 to around the 2 lakhs of the different types of the metabolites. The polarity of these metabolites varies from non-polar lipids to extremely polar inorganic ions. The functional group occurs in metabolites include alcohol, amines, organic acid, aromatic alcohol, ester, aldehyde, ether, sometimes phosphates, and in many other products. The metabolic subgroups are so large they have their own classification, such as lipidome or glycome. And also because the metabolome encompasses a relatively narrow band of molecular weight, numerous isomers. So compound, that is the concern and challenges to analysis. In the additional to the chemical complexity of metabolome, physiological concentration of metabolites in the plant tissues ranges between the millions to picomolar level. So metabolomic techniques and some applications also there. The construction of first mass spectrophotometer by the J. J. Thomson in 1912 was soon followed by a series of technological approaches, leading the first application in the ICR MS mass spectrophotometer in 1949, and the first photometer coupled with the gas chromatography was performed in the 1950s. So, although the phenomenon of NMR was not described until the mid 1940s, it was not longer before this property was being used for the structural elucidation and determination of molecules. The application of these analytical methods to metabolomics is more recent and has been accompanied by a rapid and extensive improvement in data processing technologies, mass spectrometry in metabolite studies. So MS in mass spectro, how we are to use it in the metabolomic studies. The instrumental method in the different field, especially plant and microviruses animals, which varies largely for historical reasons, but there is increasing aversion to use as many as possible for all samples due to their complementary, so as well as increasingly refined gas chromatography, mass spectrophotometer, especially those using these gas chromatography, time of flight mass spectrometry, instrument that allow is much better to conventional than by the most to GC spectra. So instruments, because they are, can be recorded spectra and the sample very quickly. The Fourier transformation ion cyclone resonance mass spectrophotometer is a very high resolution mass spectral method with mass accuracy is better than 1 ppm, which allows the separation and empirical formula measurement of potentially thousands of metabolites. Potentially holistic approach to metabolome analysis is done primarily by the recent advances in mass spectro technology and by the efforts of functional genomics research. Most technology for metabolomics is based on mass spectrum. So GCMS means gas spectrophotometer and HPLC photobody arrays. MS remains the method of choice for quantitative and qualitative metabolite profiling. The ultimate goal of metabolomics, the ability to efficiently detect and quantifying every metabolite in the plant extract, is difficulty to be attained by any single analytical method variable at present. So some metabolite selection occur in all the methodologies from initial the different solvent extraction process through chromatography to mass spectroionization. Oliver Finch's report the use of rapid scanning time of flight that is spectrocoupled with the gas chromatography separation and also integrated with the peak deconventional software. This technique increased the number of metabolites detected by GCMS in the crude plant extract to 500 to in 1000. The direct infusion of extract into mass spectro instrument using the soft electroscopy or atomic spheric pressure chemically ionization sources is an attractive way to obtain the fingerprints of the molecular ion of the metabolite present in the plant extract. So such use of demonstration, the presentation by Asaf Arboni and Dan Woodnew, the Canadian, they can the some conferences. So this powerful and alternative extensive mass analysis has the ability to generate the mass data of sufficient accuracy for the determination definitive empirical formula. Several hundred ions were observed in this for each ionization method in both positive and negative models. The metabolomics analysis is supposed to avoid the bias against certain compound classes and to allow for the analysis of every metabolite individually. Although this aim has not been reached, but the clear progress in this direction was reported. For example, a combination of different metabolite profiling tools were used by 6D Wash for the analysis of metabolic effect high flavonoid genetically modified tomatoes. The using LC photobioid photodioid arrays detection as well as LC means QTOF MS and direct infusion. It was found the flavonoid content were up to 700 fold more when they compared with those of common cultivar and glycosidic structures were presented for all the glycon intermediate that accumulated. So those analyses were performed in the tandem with the GCMS profiling of the volatile compounds using headspace solid phase micro extraction. In this, in addition to an increase in the flavonoid and methyl salicylate, a volatile compound was found to be increased in the transgenic plant over expressing the transcription factor. So nuclear magnetic spectrophotometry in the metabolomic, that is a problem. How we have to solve this? In addition to MS, means mass spectrophotometer, MNR also the play an important role in the recording the metabolic profile data for the more concentration metabolites nuclear magnetic resonance have been provided both qualitative and quantitative information. So these nuclear magnetic resonance means MNR is not sufficiently sensitive for use with the metabolite at low concentration. So another is the Marine Defarnes Institute of Food Research, and the different scientists are there in London, and another one is Arden Charlton in New York. So they presented use of proton NMR for crude plant extract, followed by multivariate analysis to cluster data sets to highlight differences. So this method of analysis gives a comprehensive summation fingerprint of all metabolites extracted and can give direct structural information regarding the individual metabolites in the mixture, particularly when the two-dimensional techniques are used. Therefore, it is suitable for high throughput, rapid first pass screening, subtraction of these data sets generate uh, virtual MNR. So it is the suitable for high throughput, rapid first pass screening. Subtraction of these data sets generate virtual MNR spectra and hence important structural data on compounds contributing to difference between samples. The Fourier transformation means transform infrared spectroscopy (FTIR), the metabolic study. The Fourier transform infrared spectroscopy (FTIR) has also been used to spectroscopically analyze the metabolome. This method detects even small phenotypic differences which cannot be measured by other methods because FTIR is chemically non-selective and it is very much suited for rapid fingerprinting of metabolome rather than identification of specific metabolite. So GC means gas chromatography metabolomic study. How the GC we have to use at the low concentration in metabolomic study, sequential approaches of concentration separation and detection method are commonly employed. So those metabolites which have vapor the pressure or can be converted into volatile products can be separated by the gas chromatography. The major disadvantage of GC for metabolic profiling is that the non-volatile compounds cannot be stored unless time the consuming the sample derivation is flight. Uh, HPLC, how we are to use HPLC in the metabolomic study. The separation of non-volatile metabolites which make up most of the metabolome concentration can be achieved by the liquid chromatography technique. So, research is high performance liquid chromatography, that one we call it HPLC, has been used for the separation of non-volatile metabolites at low concentration and varying polarities. So, these HPLC is not ideal for the rapid screening process because of its low resolution and low analysis as compared to gas phase separation. The recently ultra performance liquid chromatography UPLC has been introduced for probing due to the small diameter of packing material in UPLC and the novel silicosyl. So capillary electrophoresis in metabolomic profiling. The capillary electrophoresis can also be used in the metabolomic profiling, especially to separate cation and ion uh, metabolism, but is slow in some cases, requiring up to 16 hours for the complete analysis. So, after chromatographic or electrophoresis separation, identification of metabolites is normally achieved by mass spectrophotometer. Time of flight and quadruple and ion trap mass spectrophotometer have also been used for after chromatographic separation to identify metabolites. Another method is the electrospray, electrospray ionization, time of flight mass spectrometer, ASITOF MS, which is the generally referred to as the direct injection. MS means mass spectrophoto. Electrospray ionization is applied to metabolites containing the polar meiosis in their molecular structure.